Activity 2 is lymph organs and other nodes and vessels. So you're going to use the torso model and intestinal villi model, and I'll actually use the bust model as well to display the organs that you're supposed to look for in this particular activity. So it first says to find the spleen on the torso. Why is it part of the lymphatic system if lymph flow or lymph fluid does not flow directly through it? So on the torso model, here is the spleen here. And if you want, you can see it on this model too. The spleen is right there next to the stomach. But the spleen, why it's considered a lymphatic organ even though lymph fluid doesn't flow through it? The reason why is because even though it's not directly part of lymph fluid flow, it does still filter out our blood for bloodborne pathogens. So the leukocytes and particularly macrophages at the spleen are able to filter out the blood in order to get rid of bloodborne pathogens like bacteria or viruses or any other foreign organism that could harm us. And so because it does filter the blood for those bloodborne pathogens, and it also filters the blood for old red blood cells, we do consider it part of the lymphatic system. And then it asks, what does MALT stand for? So MALT stands for, the M is for mucosa, A is for associated, L is for lymphoid, and then T is for tissue. So mucosa associated lymphoid tissue. List all the examples of MALT tissues below. So the ones that we went over in lecture include the tonsils, which you'll have to see on this model. We actually have 3 sets of tonsils. We have the pharyngeal tonsils here at the very top behind the nasal cavity. So this little piece of tissue here, that is the pharyngeal tonsil. That is only one single tonsil. Here's the lingual tonsil behind the tongue. Again, this is just one singular tonsil. And then back here at the letter U, there's a little ball structure here, a round or spherical structure here. That is called the palatine tonsil. The palatine tonsil, we actually have 2 of, one on the right side, one on the left side. So you would have to indicate right versus left for the palatine tonsil, but not for the lingual tonsil or the pharyngeal tonsil. Another example of MALT tissue is the appendix. So I will show you on this model. You have to remove the large intestine to see it. But this little appendage, which is why it's called the appendix, is right here. So that is the appendix there at the very beginning of the large intestine. We'll learn later that this is a cecum, so it's hanging off the cecum here of the large intestine. So that is the appendix. And then I believe you can see it on this model as well. Yes, right at letter F, that is the appendix. And so the last one is going to be our Peyer's patches. Peyer's patches are going to be in the intestinal area, but the model that we will show that on is this intestinal villi model. And so this is a depiction of the wall of the small intestine specifically, because these are villi here. You will only see villi in the small intestine. So you know this is the small intestine here. And so the other part of MALT is going to be this blue structure here. This is a Peyer's patch. So on exam, if I ask you for this particular blue structure specifically, you're going to tell me it's a Peyer's patch. MALT would not be specific enough, and I would only give you half credit. So if I ask you for this structure specifically, you're going to say it's a Peyer's patch. If I asked you what collection of organs does it belong to, then you can say it's MALT, or mucosa-associated lymphoid tissue, and you should be able to spell it out. A couple other things I want to show you on this model that are important to note is, first of all, in lecture we talked about lacteals. Lacteals are there to, they are a specialized lymphatic capillary, and they are there to absorb fats from the food that we eat specifically. So just fats or lipids are being absorbed by the lacteals. Everything else will be absorbed by the capillary bed here at the villi. Other things to note is that these two here are the main vessels that are feeding and draining the small intestine, which you should already know are the superior mesenteric artery and vein. So this right here is a branch of the superior mesenteric artery here, and then this would eventually lead to the superior mesenteric vein. So those are representations of those particular vessels. And that's all we need to know for now on this model. We will get into it a little bit more when we get to the digestive system. Okay, Next on the, so we already did this on the intestinal villi model, find the large lymphoid follicle malt filled with the lymphocytes. So that is the Peyer's patch. And what the function of the white blood cells there, they are going to be surveying the fluid that comes through. So that would be through our digestive tract. So the fluid that's coming through our digestive tract, the Peyer's patches are going to be surveying that fluid in order to protect our small intestine from any particular infections. And then we also showed you the lacteal that drains the interstitial space of the submucosa that will be rich in digested nutrients like dietary fats. Specifically, what lacteals are going to absorb are things called chylomicrons, which we will get into when we go into the digestive system and a little bit more in nutrition as well. We will cover malt tissues like the tonsils and the respiratory system and appendix and Peyer's patches in the digestive system, but I went ahead and showed you now because you're going to be responsible for it for the next exam anyways. And so that is activity 2, and that is the end of your lymphatic system lab.